Hey everybody, welcome to our Exam 2 review session. So we are just going to go over the PowerPoint that I emailed out on the expectations for this week's exam. Before we jump into the PowerPoint, for the exam itself, it is worth 50 points. Most of those points are dedicated to multiple choice questions, but there are a couple matching questions in there. So once we kind of go into the PowerPoint, it should be pretty clear which ones are going to be the matching, and you should know this from the couple quizzes that you just took. So we will get started here. Let me pull up my PowerPoint. All right. So the exam itself is going to cover weeks 4 through 6. So for material, so in week 4 was all about antibiotics. These are the list of antibiotics I do ask you to know for the exam, so they should be really similar to the ones you had to know for the quiz. So make sure that with these antibiotics that you know the mode of action of each one. So basically, how do they target bacteria to, kill them? So is it by cell wall synthesis, cell membrane, protein, DNA, folic acid, RNA? So know the mode of action for each one. Also know the spectrum of activity, meaning what bacteria do they target? Do they target only gram-positives or only gram-negatives, or are they broad spectrum and target both? So here's kind of that list where it says no mode of action and spectrum of activity on those listed antibiotics. Actually, I want to go over so I can type on here. I know there was, on your guys' study guide that you did in week 4, I tried to always put comments on there of ones that I really wanted you to know, specifically one group it really targeted well. Some of you, I think, took that in. Some of you might have missed looking at that or remembering it. So I just want to really clarify here again. Most of these are broad spectrum, but there's a couple, like polymyxins are definitely just targeting gram-negatives. Vancomycin, which is part of the glycopeptides in general, target gram-positives. Let's see here. Ketolides. I know that this one is a little confusing, so because I think sometimes it's written that it can cover both, but it doesn't cover all of it together all the time. It covers mostly gram-positives. It covers some gram-negatives, but again, it's mainly targeting gram-positives, so I want you to take that away. Same with the streptogramines is gram-positive. If it helps you, the word strep indicates it's probably targeting gram-positive. Strep is a gram-positive. And metronidazole targets more gram-negatives. So the rest are kind of more broad spectrum, but those are the ones that are a little bit more specific. So I would just make sure that you note those to help you do better on that section. All right, so let's jump back into this. Okay, so other things, so that's where I have the clarification here. Ketolides state that they're gram-positive and some fastidious gram-negative. So no, they are mostly gram-positive. So that was what I was just referring to. You can see that mostly they're gram-positive. They're very select on the gram-negative. And because of that, I decided it should be mostly just known for gram-positive. And I tried to indicate that on your week 4 study guides as well. So again, similar format to the quiz that you guys took on the antibiotics in that it'll be matching. So that's where the matching questions come in on this exam. The rest of the exam is pretty much multiple choice. All right, some terms that are also out of that week 4 is the term MIC, minimum inhibitory concentration, and then intrinsic versus acquired resistance. That might be more off of your week 4 lab questions, so definitely go look at that. And then bactericidal versus bacteriostatic. So make sure that you are familiar with these terms. Look up in the PowerPoint that we used in that week. Look up your book. Look at the lab review questions that you did. Or not lab review questions. I'm so sorry. I don't know why I keep saying lab review questions. Just your book and your PowerPoint and your study guide stuff. That should get you through. Ignore what I said about the lab review. I'm not sure why I said that. So, all right. So that should get you through your week 4 material. Into week 5. Week 5 was all about Staphylococcus. Thankfully, I don't feel there's a ton to remember here. Well, at least there's a minimal amount of organisms, and these are the organisms that I want you to know for the exam are listed there in that chart. So it's just kind of having a grasp on these ones, these 5 only, so that helps whittle down what we need. So I'm going to type into this chart as we go. Let me see. I'll kind of make it bigger here. All right, so Of course, these are all gram-positive cocci. Staphylococcus are all gram-positive cocci, tending to occur more in clusters than anything else. They are all catalase positive. Remember, that is always the first test that we run. Whenever we have a gram-positive cocci, we always run catalase first. So staph are all catalase positive. So that'll be there. Once it's catalase positive, hopefully you guys know already that we're going to do a coagulase. Staph aureus, hugely coagulase positive. It is the one that is definitely coagulase positive all the time. So again, there are two different forms of coagulase tests. I think I have that details on the last slide of this PowerPoint. So we'll come back to that coagulase thing in a minute. So those are the two test results that are big. And I'll add on some others here in a second. As far as diseases go, this is just a huge pathogen. It causes so many different things that I just always say everything, because it can really cause almost anything. It goes from skin infections, you know, to UTIs, to wound infections, to septicemia, you know, to pneumonia. Like, it just goes a whole spectrum of different types of diseases. So we're not going to get too specific here. As far as other goes, we should remember it is found as normal flora of the nares, which is your nostrils, your perineal area, and some skin. I don't like to capitalize too much on that. The only other thing is there is methicillin-resistant Staph aureus, which is the organism or strain that is resistant to the beta-lactam groups. So we can usually use a vancomycin to treat that. All right, so one other thing to add on here, and we're just going to put it in the other category, is there was a new agar that we learned called mannitol salt agar. That is a selective media to screen for Staph aureus, and when the colonies show up on that mannitol salt agar, they will be yellow if it is Staph aureus. So, okay, we'll come back and add a couple things, but otherwise I think we covered the majority of what we needed to know for Staph aureus there. The next one is Staph intermedius. Again, catalase positive, as it is a staph. Coagulase, this is the one that is coagulase variable from time to time. So as a result, we can do a couple extra tests to help separate it from the Staph aureus. So PYR positive, VP negative. And then up here it'd be the opposite, PYR negative, VP positive for Staph aureus. So know those PYR VP results to help separate aureus from intermedius. And again, every microlab will do different tests. A lot of labs might not even do these, but they're just something to kind of understand how to further separate. If you do get another organism that might be coagulase positive, there are other tests that we can always add in to separate between them. All right, diseases, the main one was dog bite, wound infection. So that's it. It is found in dog saliva, which is why it can transfer over into a dog bite wound. There's not much else you need to know for Staph intermedius. That's pretty much it. All right, Staph epidermidis, catalase positive because it is a staph. Coagulase negative. This is one of the ones that we consider coagulase neg staph. So pretty much all other staphs are under the coag neg staph side of the chart. Basically, it's just Staph aureus and then maybe Staph intermedius on the coag pos side. So Staph epidermidis, catalase positive, coagulase negative, and then novabiocin susceptible, or you can term that positive, if you will. All right, as far as diseases, there was only one disease we really linked with this, and that was medical devices. So this is the one, it's heavily found as normal flora of the skin and mucous membranes. That's why it's named epidermidis for the skin. So it's hugely found on our skin normally. As a result, a lot of times it's just contamination in the microlab, because if they don't do an aseptic collection process, say of a wound, and they pick up a little bit of the skin, we could grow it Staph epi then on the agar. So a lot of times it's just contamination, but when it does cause disease, it's typically linked to those medical devices. And again, it's because it can produce this exopolysaccharide slime. I can't spell today. There we go. So it can create this layer of slime that really adheres it to the prosthetic valves, the medical devices, catheters, you know, in general. And then when they go and put those into the patient, it gets introduced into a sterile area, and then it's like, oh, we're here. We can do what we want. So it causes infection that way. Otherwise, pretty much just shows up as contamination. And then Staph saprophyticus, this is catalase positive, coagulase negative, novobiocin resistant. Pretty much the only one that's novobiocin resistant under the coagulase negative side. All the other coagulase negative staph tend to be novobiocin susceptible. So this will help separate Staph saprophyticus from all the other coag neg staphs that are out there. So it kind of keeps it unique. And then again, pretty much just UTIs. We don't really see this outside of the urinary tract world. It only really is associated with UTIs. And that is because, again, it is found as normal flora in the genitourinary tract. So it typically affects people women especially that are having sexual activity, and if they don't go to the bathroom, urinate, and cleanse well afterwards, they allowed this bacteria maybe to get pushed further into the urinary tract and then cause an infection. All right, so that should take care of that. And then finally, Micrococcus. Very similar to Staph, but it is its own genus. It is catalase positive, coagulase negative. This is the one that's pretty much rare to cause any sort of disease. It is always contamination. I wanted you guys to still know what it was, though, because it will pop up as contaminants in the micro world. And so instead of you being like, what's Micrococcus? I've never heard of it. At least you've at least heard of it at this point. So this is the one that has some really good, fun looks to it. It can be almost any colony morphology: white, pink, orange, tan. You know, so it kind of some fun looks on the agar plate. But otherwise, we're not going to do anything with it. It's just pretty much contamination. Extremely rare for it to actually cause a disease. All right, the only other thing I think we're missing here is, let's make sure that we know the colony morphology of Staph aureus. You guys have all seen this in the lab now. And again, when I talk about colony morphology, I'm talking about the typical classic look. There is always exceptions. Nothing is ever picture perfect. You're going to see exceptions to all this, but these are going to be the typical examples with each one. So just keep that in mind. So Staph aureus tends to be beta hemolytic. It can sometimes show up as non-hemolytic, but it tends to be beta hemolytic, medium-sized colonies. Sometimes it may have a slight yellow hue to it. Again, aureus stands for gold, so it can sometimes have a slight buttery yellow look to it. Other times it's just white. So it just depends on the strain you get. The other one I should think you really should know is Staph epidermidis. This one is usually gamma hemolytic, so meaning non-hemolytic. And then again, it tends to be medium white colonies. So you should have seen that in a lab as well this week. So there is your staphs kind of detailed out. So if we screen back here, if you're listening to this as a recording and you didn't get a chance to write everything down, pause the recording and write everything out as you see it here. I am going to move on, but again, go and pause the recording. That's the beauty of a recording. All right, and then week 6 was all about Streptococcus and Enterococcus. So let us go back and zoom back in. All right, the thing about Streptococcus is first that they have different Lancefield groups assigned to the organisms. So again, Lancefield groups are based on carbohydrate antigens on the surface of that bacteria. Some have these specific carbohydrate antigens and they were named for it. Others don't have them at all and therefore have no group. So make sure that you kind of have that understanding of what a Lancefield group is. And in fact, there are kits in the lab that will do a Lancefield group testing to screen for those carbohydrate antigens. There's actually kits made to target and determine which carbohydrate antigen is present. We can use those as well to help identify streps. So strep pyogenes is very much known as group A strep. So that is the carbohydrate antigen on its surface. You should know the names interchangeably, whether I say group A strep or whether I say strep pyogenes, you should know they're the same thing. The other thing with streps, of course, is once we know it's a Gram-Pos cocci, and again, streps tend to occur more in chains or pairs for Gram-Pos cocci, then you catalase test it, and they should all be catalase negative. After that point, you're going to go back and look at your agar plate and look at the hemolysis. Hemolysis is so essential in the world of stress. It helps point to the next set of tests that you're going to do. There's a lot of different tests that we can run, and we don't want to run them all just to run them. We want to have kind of a plan. So once we know it's a catalase negative Gram-Pos cocci, then we look at the hemolysis, and the hemolysis will point to the next set of tests to run. So in the case of strep pyogenes, it is very much beta hemolytic. So in that case, I could run a bacitracin A disc, and it would be susceptible to that disc. I'm running out of room. I'm going to roll this over. There we go. We could also run a PYR, and that would be PYR positive. And you guys, I know, just filled this out on your study guide, and so make sure that you know those test results. I tried to add on to the study guides, or I saw something might have been missing that I wanted. All right, it is CAMP negative and hip urate hydrolysis negative. So those are a good representation of some beta hemolytic tests for strep. As far as diseases go, strep pyogenes causes a lot of diseases. It's like Staph aureus in that it's always a pathogen. It's never normal flora. It might be carried by some people, like in the upper respiratory area, but is not ever considered normal flora of that person. So just wanted to mention that there. So of course, we all know this is the main cause of strep throat, what we can call pharyngitis. So that one I think we're all familiar with. Otherwise, there is a ton of things. There's different skin infections like impetigo, just skin infections in general, and then there is the necrotizing fasciitis, which is that nasty flesh-eating disorder. But again, Staph aureus can do that too. Toxic shock syndrome, which Staph aureus can do as well. So very similar things in that regard. Scarlet fever, which we don't really see a lot anymore of. So the list kind of goes on. And then there's those two that we call post-streptococcal complications, meaning that after you recover from a strep infection, your body has made antibodies to fight the strep infection. And those antibodies will bind up with the strep antigens, and help destroy it. Well, what happens is that antigen-antibody complex can also do damage to some organs like your heart or your kidneys in the process. And that's why we call them post-strep complications. So after you've recovered from maybe a strep infection of some sort, you might result in getting one of these two, which is rheumatic fever, which involves the heart, or acute glomerulonephritis, which involves the kidneys. And it's all due to those antibody complexes made as part of fighting the strep infection and thus causing damage to the heart or kidneys. So those are the two post-strep complications. So again, the list of diseases is vast. There's a lot of them, but this is a nice representation of what we could see. And I just lost my thought of what I was going to do next. Oh, I remember. While we're on the topic of strep pyogenes, in your study guide this week, you guys should have been doing virulence factors for strep pyogenes. I did ask that you know the virulence factors for strep pyogenes. So remember, there's M protein. That is a protein in which you have to have in order to make the rest of the virulence factors that help it cause disease. There's the hyaluronidase factor, which is known as a spreading factor. It can help the organism spread through the connective tissues. DNase, there's different DNase enzymes that it can make to help target and destroy DNA of your phagocytic cells, so that it can get rid of them. It has streptolysin O and S, which help make the beta hemolysis on the agar plate. So there's streptokinase, I think that's it for now. But know those, again, you guys all define those on the study guide for week 6. So go back and look at your definitions or look at your book or whatever you need to help remember these virulence factors for strep pyogenes. All right, I'm going to lower that back down. All right, let's do our next strep, which is strep agalactiae. This is known as group B. It is a beta hemolytic strep. So strep pyogenes and agalactiae are two main beta hemolytic streps. Again, it is a strep, so it is catalase negative. And we can run the exact same test because it's a beta hemolytic strep. We're going to run those beta hemolytic strep tests. So bacitracin A disc resistant, PYR negative, CAMP positive, and hip urate positive. So you can see how those tests will tell us which one of the beta hemolytic streps that we have. This one is simpler in that I only asked you to remember one disease with it, and that was neonatal meningitis. So this is the one that women often get screened for during their pregnancy to see if they carry it in their vaginal tract. If they do, they're given antibiotics to wipe it out. That way, upon birth, they don't pass it to the fetus and it results in this meningitis situation. So I always say group B for baby. It's all about the baby with strep agalactiae. All right, Strep pneumoniae, this is one of the ones that has no Lancefield group, so nothing there to worry about. It is an alpha hemolytic strep. Again, it is catalase negative as it's a strep. And then let's do our alpha test, which would be Opticin, or you can say Opticin, which is the P disc. It would be susceptible and biosolubility positive. So those are a couple really good tests to identify Strep pneumoniae. And just like the name implies, it causes pneumonia. It's actually the #1 cause of pneumonia, and it causes meningitis. So two nasty infections there. This is also the one that is a gram-positive diplococci. So what we can say GPDC for short. So it occurs in pairs, and those pairs are said to be cat-eye or lancet shaped. So kind of the pointy ends on those circles. Even though they're supposed to be round cocci, they have kind of pointier ends to make them that cat-eye shape. All right, viridin strep, again, no Lancefield group, alpha hemolytic. So let's do the same test. It'll be still catalase negative, Opticin, P disc, resistant, and biosolubility negative. I cannot type. Okay. For diseases, this is in a ton of different alpha hemolytic streps, all categorized together under the viridin name. These alpha hemolytic streps don't tend to cause a lot of infections, so we just kind of group them under one title. But when they do, it typically is like endocarditis or dental caries type infections. All right, Enterococcus faecalis has no real Lancefield group name. Sometimes you might hear it referred to as group D, but there are other group D streps out there, so just kind of a note there, but typically no Lancefield group. Can be any hemolysis, so this one is a little trickier. Catalase, weak positive. It might be so weakly positive that you don't really see any bubbles at all and call it negative. That's okay. For tests, there's going to be a few different ones unique to it. It is PYR positive. It likes to grow in 6.5% NaCl, so it loves itself some salt solution there. It is bioesculin positive. It is biosolubility negative, though, so that'll separate it out from Strep pneumoniae. So there is some good representation of a few tests that will help identify it. As far as diseases, it pretty much is just associated with nosocomial type infections. Again, nosocomial referring to hospital-acquired infections. So a good one that we see it pick up a lot with is UTIs, so a nosocomial UTI. All right, so that should be your Enterococcus and the rest of your streps. So again, if you didn't get a chance to write everything down, pause it, write it out. Look back at your week 6 study guide on this. You guys filled all this information out there. I tried to add in comments here and there of ones I thought you were missing or things that I saw, but otherwise, you know, look at the PowerPoint, look at your study guide, use this, look in your book if you need to, and you guys should be just fine. There's only really, again, 5 organisms here to remember. There might be more test results to remember, but there's only really 5 organisms to remember, so not that many. And then finally, overall, we also discussed catalase and coagulase. As we just saw, we've been using those a bit in the lab now. So again, the purpose of a catalase test is to separate between whether you have a strep or a staph. Staph are catalase positive, whereas strep are catalase negative. So you guys should have all been doing that now in lab, and that should make more sense to you. So that is always the first test that we run after we have a gram-positive cocci. That way we know if we're dealing with staph or if we're dealing with strep. Again, as you guys did in the lab, you should have known the reagent is hydrogen peroxide, otherwise known as H2O2. So that hydrogen peroxide, you just put a drop in, mix in your organism, bubbles, it's staph, no bubbles, it's strep, or maybe it's a weak pos Enterococcus situation. Simplest test ever, love it. I love that catalase test, it's so simple. If it is catalase positive, then you do that coagulase test next, just like we did on the staph chart there. So again, the purpose of coagulase is to differentiate between Staph aureus, which is always coagulase positive, from your other staph. There are 2 types of coagulase tests. I'm thinking that most of you guys probably did the slide one where you looked for agglutination on a card. That's a really common way of testing coagulase. So the slide one is looking for the bound coagulase enzyme. If you were to do the tube coagulase test, that is done in a tube. It has to be incubated for a while, so it's not as quick as easy to do. But it's looking for the what we call free or extracellular coagulase enzyme. And again, Staph aureus will produce both types of coagulase enzyme. So some labs will only do one coagulase test. Some labs might do both and use one as a confirmatory method for the other kind of situation. So that's just a bit of notes on that. All right, so that kind of is it for weeks 4 through 6. You guys do unfortunately have some memorization ahead of you for those antibiotics and all the tests and stuff, but it's doable. I think it's a learnable amount, especially since we've been answering questions on these kind of over and over again. So use your study guides from the various weeks, look back at the original PowerPoint, things like that, and you guys should do just fine. If you do have a question for me at any time, please feel free to email me. Otherwise, have a fabulous week, you guys. Thank you.